I'm Tammy.
I'm Jalen.
I'm Lena.
And I'm Sai.
And this is our experiment. So basically some background on this whole experiment is based off diffusion. And diffusion is when particles are in a high concentrate in one place and then they distribute to a place with low concentrate, which means it doesn't have any. And it continues to do that until it's equal, so that's called equilibrium. And in this case, the water had the highest concentration compared to the egg because the water is the one with a dye in it, and then the egg had no dye. And so what we were trying to do is get it to go to equilibrium and have the dye distribute into the egg. And our hypothesis was based off temperature, and our hypothesis was that if we add heat to the egg, then the particles will be caused to move faster, which will mean that it will distribute more evenly into the egg and have it just more into depth. And if it is cold, then it will be slowed down, and then there's probably going to be more light in the middle.
And our independent variable for this experiment was the change in temperature. Our dependent variable was how much dye got into the egg. Some controlled variables was the size of the egg, the amount of water, and the amount of dye.
A was our control group, and B and C were our experimental groups. And A, we just had it shown on the counter at 27 degrees Celsius for room temperature. Then B, we put on a hot plate and heated it to 40 degrees Celsius. And C, we put in the refrigerator and it cooled to 12 degrees Celsius.
Our results were that when we added heat to the egg, the particles began to move faster and it dyed the egg more in depth. But when we added it to the colder temperatures, the particles began to move slower and did not dye the egg as much. So by that, our hypothesis was correct.
Some ways we can make this experiment better is by testing more temperatures and just getting more tests and results to make our
I'm Kayla.
I'm Sydney. I'm Maisie.
And this is our diffusion experiment. The diffusion is the process of particles moving from an area of high concentration to an area of low concentration for equilibrium. Isotonic is concentration is equal. Hypertonic is the particles leave the cell and it shrivels up. And then hypertonic is they enter the cell and expand and explodes.
Our hypothesis was if we mix a solution that it diffused further into the egg.
Our independent variable was the speed of mixing. We had one group that was at the speed of 1 and one that was at the speed of 3. And our dependent variable was how far the red dye diffused into the egg. We had several controlled variables. One was the size of the beaker. One was the amount of water, the amount of food dye, the amount of egg. The control group was group A, and the experimental groups were group B and C.
With our results, you can see that all of the red dye diffused through every egg, so it went all the way through. So our results aren't as clear as they should be.
Our conclusion was that our conclusion showed us that the egg had fused all the way through, and we couldn't tell much of a difference. We thought mixing would help the particles move further into the egg. Diffusion is movement, and particles would move faster, and diffusion would move in further.
One suggestion to make our experiment better next time would be to cut the pieces thicker so that our results are a little more clear.
My name's Steven.
My name's Carson.
My name's Tia.
My name's Michael.
And this is our diffusion project. What we did is we changed the size of the particle, and as you can see, the red dye had red 40 in it, which was 496.2 grams per mole. Blue 1 was in the blue, and it had 792.68 gram per mole. And yellow 5 was in the yellow dye, and it had 535.3 grams per mole. It really showed off diffusion what diffusion is that it takes an area with very high concentration and it kind of like leaks through the membrane into an area with low concentration. So in this experiment, the high concentration area was the water with the food going in it, and the low concentration was the egg. and as the 24 hours went by, it soaked into the egg and created diffusion.
Our hypothesis was if you change the particle size, that the red dye would go through the egg better.
Our independent variable was the dye. We had red, blue, and yellow. Our dependent variable was the amount of absorption the egg had from the dye. Our control variables were amount of dye, amount of water, type of water, the container size of eggs, type of eggs, and the temperature. Our control variable was red because it had the same particle size. Our experimental groups were blue and yellow because they had different particle sizes.
Alright, so our results were that red had the dye leak through the most. Red dye was most, yellow was not far behind it. Both showed about the same amount of white in it. Then blue showed the most amount of white out of them all. So our conclusion was, of course, red had the most diffusion in it because red had the least amount of mass in it. And when you have little amount of mass, it's easier to dilute. Unlike
Things that we could have done, right? We could cut like bigger pieces of the egg so it would soak through as easy.
I'm Josh.
I'm Carter.
I'm Garen.
I'm Zach.
And this is the amount of food coloring. Diffusion is the process of taking particles or ions and moving them from a high concentration zone to a low concentration zone.
Our hypothesis was if we add more food coloring, then it will diffuse faster.
The independent variable was the amount of food coloring that we put into the container. The dependent variable would be the rate of diffusion. Some control variables would be they are all in there for 24 hours, the amount of water and the container that they're in. The control variable would be the 0.5 milliliters. The first experimental group would be the 1 milliliter, and the second experimental group would be the 3.3 milliliter.
Our result was that group B will diffuse faster because it had higher concentration.
We got these results because we figured since the egg would be represented as a selectively permeable membrane, that since the food coloring particles are so small that they would just diffuse right into the egg.
A better way to improve on this experiment is using better equipment and cutting the eggs more evenly.
The presentation is brought to you by Drew Salads,
Peyton Runke,
Elliot Jensen,
and Ben.
Okay, so here on our left diagram here, we have concentration gradient. And on the right, we have equilibrium. Now, over time, as you can see, where it says time here, we have concentration gradient will turn into equilibrium. So here on the left, we have our red food coloring liquid and our egg on the right. Now, over time, like I said previously, that will turn over into equilibrium. Now, equilibrium is when it's equal on both sides, and concentration gradient is when it's not equal on both sides.
Knowing this, we can apply it to our experiment by figuring out that over time the food coloring would move into the egg. So what we did is by changing the size of the egg, we figured that the time it would take for it to diffuse into the center would be a lot shorter. So our hypothesis was if we decrease the size of the egg, then the percentage of the egg colored would be larger.
Our independent variable was how thick we cut our eggs. Our dependent variable was how far the food coloring traveled, and our control variables were such as light, temperature, and how concentrated the food coloring was in the solution.
We had three groups. We had our control group, our first group, and our second group. This one is named Robin, this one is named Vulture, and this one is Godzilla. Our first control group was cut 1 inch thick, or 1 centimeter thick. Our first experimental group was cut 0.5 centimeters thick, and our second experimental group was cut 1.5 centimeters thick.
Our second group, we had 0.25 centimeters of diffusion. And our third group, our last group, we had 0.5 centimeters of diffusion.
Now, from these results, we can confidently say that the red food coloring diffused into our egg at the same rate. Now, the reason why it's 0.25 on our second egg is because it's only 0.5 centimeters thick. So it only took 0.25 centimeters to reach the center, but it was consistent with our other two groups. Now, from this, we can say that our hypothesis was correct because the smallest egg that we cut right here, it took the least amount of time to reach the center, whereas with these two, we had enough light to still be able to measure it at 0.5 centimeters. This could help explain why cells are the size that they are. Because they're so tiny, they can easily diffuse particles through them. This is why the cell of a mouse is the same size as the cell of a dinosaur. If the cells were any larger or smaller, the speed at which the particles would diffuse through the cells would work a lot differently and might not get to the cells that need them the most. Some ways we could have improved this experiment was by exposing the egg to red food coloring maybe on only one side. That way we could more accurately measure how far it went into the egg. We could also help that by having more groups or increasing the time or the size of the egg. This way we'd be able to calculate the exact rate that the food coloring went into the egg, and therefore we could compare it better to see if our hypothesis was actually correct.